So the first question I want to ask our panelists is which technology is going to have the greatest impact on development of novel treatments for human diseases in the next 10 years? So this can be in your field or outside your field. And Simone, why don't we start with you?
Thank you very much, Andrew. Well, I think it's not one technology, first of all, and I think treatment starts with diagnosis. And so I think here we see really remarkable developments in wearables, in point-of-care diagnostics using microfluidics, analyzing saliva, sweat, urine at home, right, to basically identify earlier when a person gets off track. And then the next step is basically a visualizable medicine that we heard, right, imaging. I think we see tremendous progress here on how we can actually have sustainable and affordable imaging methods where, for example, in my group, we leverage ultrasound by designing molecularly responsive ultrasound contrast agents. We saw examples of how we can push sensitivity and specificity, right, to basically get better at the diagnostics, to then really identify what treatment is needed. And then here, and we saw this also in the keynote speeches, of course, then in the treatment development, in drug design, in drug development, we see now how AI is, of course, supporting, right? So I think these are the big technological developments, how robotics, of course, and automation helps us then to do high throughput screening, and then combined, and here we come to the surveying, by getting better in targeting, both short and long distance. So short distance targeting is all what we've heard in on protein interactions, ligand interactions, and getting better on, hooking up to a cell with the drug. And the long distance targeting is then by robotic methods to really help accumulate certain drug carriers with the drug at the specific site to enable these short distance interactions. So it's a convergence of disciplines and technologies that I see for the next 10 years coming together.
Tyler, what are your thoughts on this?
I agree completely with what Simone was saying. But
Move the microphone a little bit.
I would agree. I would say the biggest thing in our field in medicine is going to be the AI. I mean, this conference, and specifically looking at the structural component of biology and what that's going to open up in terms of DNA, RNA, and the protein structures. If we can use and leverage AI to look at protein structures and understand them at the molecular level, which is going to be happening in the next 5 to 10 years, it's going to open up a completely new world for precision medicine. Less than 15% of all clinical trials succeed. So that's 1 in 8 drug makes it through the whole process. If we can even move that a few percentages by, that's going to have a huge impact in just how we're able to treat diseases, then also financially as well. So I feel like the combination of our experience and our knowledge of protein structure and the way to simulate that using AI is going to revolutionize a lot of the field.
Greg?
If I were just looking in something focused like oncology and development of what takes the most time, I think most of us would look today and say that, the advancements in minimal residual disease is probably something that will have the greatest impact about the element of drug development that takes the longest, particularly in oncology, of allowing us to move upstream. But if I really step back and look, let's be honest, time is the most important thing for the questions we wish to ask. And, when you're 5 years old, if you can remember back, 1 year seemed like a lifetime, 20% of your life. By the time you're 50, it's 2% of your life. The sun blinks an eye, though, and half the human race has come and gone. So I do think when we look at the context of machine learning in general and AI, that we do have an ability to leverage this. It's not going to answer our questions, but it can do certain things faster.
So I wanted to point out that the time from discovery of the basic science for the drug-eluting stent to FDA approval was 3 years.
Yeah.
And it's been 25 years since I discovered the leaky ryanodine receptors and the small molecule, and we're still working on that.
There's no question that time for basic research is an investment.
No.
You need to think this is a 30-year horizon to answer the important questions.
Yeah.
And, I think generally speaking, we need to remember that curiosity-driven research, it's not just the aha moment that leads to the development of a drug. It's the smart observation that helps people develop something later with that knowledge. Even if it didn't work, it stops people from doing something that would later fail in development.
So Tyler, you mentioned AI. Will AI replace scientists?
No, it will not. So what it will replace is AI or scientists who are able to leverage and use AI effectively to scale and advance their own research, and scientists will replace scientists who do not. That's what that's what will be replaced. But I would say it's a tool, and just like any other tool, it requires context. It requires certain things that it cannot do on its own. So it's not ever going to replace completely, but those who utilize it most effectively will replace those who do not.
So Greg, you're a CEO of a company making therapeutics. How much are you relying on AI, and are you replacing workers with AI?
I'll put something in a bit of context. Mm-hmm. When it comes to any AI, we're going to call it that algorithm, it's much like, the qualifying exam of a graduate student. You drill down until you find it's hallucinating, and you keep going in different areas, and you say, all right, there's one area here that something works. If I look at where we're using things that have gone from basic big data, where you say I have a 7 million diversity library of looking for a new signaling pathway. In 2015, 2016, you were 400 scientific FTE years to get through 7 million diversity libraries, which used lots of great tools. The question now is, you want to go in and take that 7 million true data set and train it with machine learning, is to take a really complex thing, which is the Johari window of 3D protein structure modeling, which is transmembrane proteins, where we say, can I actually go through and bootstrap that lack of knowledge by actually going in and training it on functional data instead of crystallography, which we don't get often from transmembrane proteins. Yep. So I think in that area, it's something that it's not eliminating jobs, but it is something where you're learning how to do new things with data you've already developed.
So Simone, you're a robotics expert. This is one area where AI is going to rule the world, I would assume.
Well, yeah, but I actually would like to answer the question first on whether it will exchange the scientists, because sure. I think just to add to that, I think it will just radically change how we work as scientists, mm-hmm. right? And I think, so my firm belief and hope is that it will give us more time for thinking, and we're coming back to actually curiosity-driven research, because we're still the ones asking the questions. And so I think it will be very crucial now to really educate and teach the next generation that grows up learning with AI to still do the hard work of deep learning and understanding, because right now we're in a position that we can still judge very well and identify the hallucination, right? Yeah. But that is a key point, because we're in the end the ones then taking decision and doing actual implementation and physical transformation, right? So that is just to add on that. I think that's a huge responsibility we have right now also for our next generation. And then in terms of robotics, of course, I mean, we've been using a lot of AI already, and it's just unleashing basically now new levels of just in terms of speed and accuracy, and I think in that sense it's something we've been long anticipating.
So just to follow up on this discussion about how to make new therapeutics, maybe Greg, we'll start with you. What is the current biggest impediment to delivering new therapeutics to patients in your field, and how can it be overcome?
I think, look, the time it takes to get from molecule to medicine, half your life has been worked through. But I think what we've done with the opportunities today is to look at the element of being able to optimize a therapy in early clinical development rather than making each molecule as a scaled product, is thinking about them as prototypes. When you would look at something of a therapeutic going in first in human and say, should that affinity be higher or lower for a particular modality, particularly with complex multimodal therapies where we say we have a goal to say, can you CR without Cy/Flu or the ICU? There's 10 years of things going into a platform. But I think the element is, if you take each one of those and assume that you're going to go in and develop each one as scale as a product before you know that that's the optimal product, I think what happens is you can spend far more time than necessary. And I think for many of the things of the next generation of medicines, we have to learn how to collaborate with our clinical colleagues and do translational research. And because things are becoming faster, to say, I'm not going to test one moiety in the clinic, but rather 3 or 4, and say, let's actually use a Bayesian design and say, which one is the proper medicine to really invest for the next 10 years to actually make a difference.
Yeah, that's kind of anathema to big pharma, I would think, these days, right?
Yeah.
You go for the best molecule after you. So Tyler, I know that you're probably far away from a therapeutic application, but I'm sure you've thought about, and cancer being such a difficult challenge. Where do you see that, barriers being, and how are you going to overcome them?
Yeah, I would agree with everything that Gregory said. It's a lot of it's the time, and then also, like I mentioned earlier, it's the failure rate. A lot of these drugs or these therapies do not pass or make it through clinical trials successfully. And so that's a huge barrier. If you have all of this time, effort, and like you said, a lifetime's worth or, several decades sometimes of research and work and millions of dollars, sometimes even more, going into this research. And then the end of the day or the end of the life cycle, it doesn't even work or pan out to a actionable item that you can use in the real world, then that's, a lot of wasted time. So I feel like we mentioned, we were saying with AI and the advent of some of the emerging technologies and precision medicine that is really going to personalize where we are at the field, it's going to be really important for us to be able to look at what we're studying and then do it in an accelerated fashion where we're not spending so much time, but actually doing things more efficiently so that things get to market quicker, and then there's a higher success rate. And, it's never going to be in the, 90s or 80% success rate. That's simply not possible. Greg can probably talk more about that, but I would say if we can even get it percentages higher, that will save a lot of time, a lot of effort, or help a lot more people, and be much more lucrative in terms of the, industry capacity.
So Simone, where are nanorobotics going in all of this for therapy, and what do you see as the real challenges, and how are you going to overcome them?
Well, in therapy, I really see the delivery issue still huge, right? We have, as I mentioned before, under short distance targeting and interactions, we have fantastic solutions at hand, what we can do with CRISPR-Cas9, for example, in genetic engineering, but then delivering basically these vehicles targeted to a local site is still challenging. And so we have, because of, the high failure of phase 3 clinical trials, because of the inefficacy at the acceptable dosage, right? So we need to get better in delivering effectively the dose to a targeted site. And so I believe that it's really then through external energies. So while in my group we focus on magnetic fields, not for imaging, but actually for guiding magnetically responsive nanocarriers that incorporate then the drug and components to bring to a site. And I do feel it's going to be, to me, again, a convergence of science, is really different disciplines coming together, where then in the end really external energies may be magnetic fields, or we have focused ultrasound. There's lots of different technologies coming together. That's really from the robotic side, because there's imaging, feedback, and in the end a closed loop control.
So what power are you talking about with the magnetic fields, and is this something that, if you have like an artificial shoulder, for example, are you not going to be able to get this therapy?
Sorry, the artificial shoulder?
No, I have like a piece of metal in here.
Oh, I'm sorry.
It's an MRI thing.
We are not working, it depends on the approach, it's not a high frequency, so there would be no heating, no eddy currents, and so there would be this compatibility, but yeah.
Our keynote speakers from today, questions, things that you would like to comment on, add to, if we can get a microphone for, okay, here we go.
Hi. I would like to know from Dr. Frost what he thinks CAR T therapy will look like in 5 years.
Medicine moves slowly. So if we're talking about commercial therapies that have reached patient access, you may have a transition of things from late-stage development between both LMP-based therapies that people are looking at in most areas like autoimmune reset, where you have a short duration. I think that autologous therapies will still have a place in certain settings, particularly in TIL-based therapies, for example, which are more neoantigen-driven. But I do think from the standpoint of patient access, that if we are smart about what we do, of learning from the past, we'll build the types of products that will get off the shelf of a transplant ward to the community, which will make them accessible. But, you know, we still have a lot of basic things about understanding cancer biology we need. And this is, if anything, a plug out again to those of asking the piece, which is for basic curiosity-driven research, particularly in cancer biology. We don't often talk about curiosity-driven research in cancer biology, but the reality is the wealth of a nation is defined by the number of scientists that can support doing basic curiosity-driven research. And these are fruit that are not harvested many times for 30 years down the road. And we can't be just thinking about the patients that we need today, because there will be patients 20 years from now and 30, and it will make us smarter in the medicines we make.
Question over here.
Not a question, a comment again, the future of cancer therapy. So you are right that AI will speed up things, but it's a tool, as you said. So,
yes.
It's not enough. And the problem with cancer, as was pointed out, is the resistance. That is, you have a wonderful drug, but after a while, the cancer cells will get wiser and will get around it. So you need a lot of new targets.
Yes.
A huge number of new targets in order to treat cancer, because the second one will also develop. And then the way to get new targets, as I say, is basic research again. So I'm coming back to this.
Yes.
So questions or comments from the audience on what we've discussed today? Please say your name.
I'm Hartmut Michael from the Max Planck Institute of Biophysics in Germany. We work on membrane transporters, and we realize that AlphaFold continuously fails. So I think your statements were exaggerated. The failures happen then when your protein undergoes a conformational change during action. And for instance, even already binding of the substrate sometimes causes substantial conformational changes which are not recognized by AlphaFold. And what you actually get is mixed structures which are neither the apo form nor the form with the complex. You still need the experiments, and you should not forget that.
You always need the experiments, absolutely.
Agreed.
Other comments? Yeah.
Just one other point with regards to resistance. And this is something we experienced very early on in solid tumor targeting and going after specific antigens, which was that you could CR the target out of a tumor, which is the most frustrating thing you can do in development. But I do think there are elements, even on the in vivo gene modifying therapies, of leveraging tumor neoantigens, we call it CAR-TIL, and actually leveraging that polyclonal response to be able to go through and get an additional value. So, but again, making it accessible is something that requires some interesting engineering.
If you have questions, don't give up. I'm going to move on to my last question for the panelists, and this is also for everybody. Do you have an idea for a new model for stabilizing funding for basic and translational science? And Simone, why don't I start with you? Because you come from a different model. Now I want to hear.
Yeah, that's a very easy one, right? No, it's a very tricky question. But I know it's a new model. I don't know how we would sustain it, but I think we need to go to more long-term funding so that we can get out of the cycles of grant proposal submissions, right? That we have a more continuity, stability. I think that actually also helps creativity, and we don't lack novel ideas, right? But it takes time. It still takes time, although we get faster. I think we also should have more cross-border and cross-discipline initiatives. I feel it's still difficult to have international collaborations to some extent, or having the right funding instruments. And yeah, so I think these would be 2 points to focus on. And then we could still have for more applied translational projects, shorter sprint type models that is then really to show whether it works. But we need longer term funding ahead.
So Tyler, you're a younger generation than I am, that's for sure, and you have a long way to go in your career, and I'm sure it's going to be amazing. But what would you like to see in terms of stabilizing funding for basic science?
Yeah, I agree. I was going to say long-term as well, because that's really key. But in addition to that, I would say we have to find a way to make basic science sexy. Like, translational research, correlative science, it gets a lot of push. There's a lot of background. There's a lot of publicity with that type of research. But basic science, like we've all been saying, this session, this is really where it starts. This is really where the fundamentals of everything that comes afterward are, developed. And we need to make it attractive. We need to make it appealing. And we have to really, I would say, shape or reframe the way that we are even thinking about it and presenting it to the public. Because it's not just, basic science as we're studying this and we found this. It's a transformative something that we have the implications that, far-reaching beyond just the one implication that it might have found in the lab. So we have to really find a way to make it, much more appealing and attractive and sexy.
Well, certainly, and I talked to Roger about this, the idea of publicizing this meeting, I think, to the public. We don't do a good enough job at advertising what we do. Greg, is there a role for industry in all of this?
Look, I think when it comes to pure curiosity-driven research, that we used to have industrial postdocs and people would be confused where I said, you don't understand. These people have to work on a hypothesis. They're not going to be on your project team. And you had beautiful work could be done there, but it's a very small fraction of all the work that can be done. And when it comes to the element of how do we make this sustainable, I think you need to get into a communication, I think, with education, with governments around the world, which is this concept of you have investors go in, you have 30-year treasuries, right? You have short-term bonds. These are 30-year treasuries. Don't expect to get your principal back at year one. That's not how it works. But I think there are some metrics that we can use of this to help quantify what's been done in a retrospective fashion. Go back and look at all the great research that's been done and use that as a metric, and getting more funding to younger generations, realizing that they have a lifetime ahead of them to build something, and making that investment where, yes, the bar may be higher, but actually pushing that out and say the people that are committed to do that, don't go drag them into industry or have them do something else. Make it a secure enough environment where they can really invest to make that difference.
So we have about a minute left. Do any of our keynote speakers want to weigh in on this topic of stabilizing funding for science? Yeah, there's a question here. Microphone.
Okay. So I think relying on the governments for funding basic research is not that robust, because when they have financial problems, the first thing they cut is the funding for basic research. So another scenario may be applying like a very small percent tax, like 0.1% tax for basic research on companies that exceed a certain threshold of revenue. So if there is a big company exceeding a certain threshold of revenue, just put 0.1% tax for basic research. And then this can be something sustainable.
Okay, well, I would like to thank our panelists and also the remarkable keynote speakers, the organizers, really exciting topic. So thank you, everybody.
Thank you. Great comments.